Hello and welcome to a brief introduction into silencing RNAs or siRNAs. And so what silencing RNAs are is they are a type of regulation that can occur within transcription to translation. So they affect the messenger RNA that is created during translation. So firstly, we have to make some messenger RNA. We're going to get those transcription factors, have them bind, get that RNA polymerase to bind, and we're going to go make our strand of messenger RNA. And so once we've made this strand of messenger RNA, we have our messenger RNA. We're going to get our silencing RNA that is injected from outside the cells, be it in viruses or molecular biologists could use this as well. And so we're going to get our silencing RNAs, and then they're going to come on in, and then there's going to be a protein called dicer. And what dicer is going to do is it's going to cut up this this silencing RNA. It's going to cut it up into small little pieces. It's going to like snip it off. It's going to cut it up into a bunch of little pieces. And so what these pieces are going to do is that now that we have these little pieces, this mini silencing silencer RNA is going to go and it is going to go bind to a protein called RISC. And so what it does is we have our silencing RNA is going to go and is going to go and is going to find a messenger RNA with the identical base pair. So it's going to go base pair, and it's going to go find a messenger RNA with an identical base pair. It's going to go scan, 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 and when it finds it, boom, we're going to get RISC to destroy this mRNA. We're going to get to destroy this mRNA, and if it makes another mRNA, it's going to keep going. We're going to scan it, we're going to find this mRNA, and we're going to destroy it, we're going to degrade it. And so why would we do this? This is this is just another way to regulate gene expression, and so this is very useful because we're able to regulate gene expression without changing the actual gene. So the gene itself is the same, nothing from the gene is going to change, nothing in the way the transcription factors come on is going to change. RNA polymerase is always going to come on in the same way. Everything is still going to get made. However, this is a really nice way to add layers of complexity. So we change the gene expression because we're not translating, we're not making those proteins that go out and do specific functions. We're changing all of that, but we're not changing the actual gene. We're keeping that the same. And so that is a brief introduction to silencing RNAs.